Electrolytes are essential minerals in the blood and tissues that carry an electric charge. They include calcium, carbon dioxide, chloride, potassium, sodium, magnesium, and more. And these ions play crucial roles in maintaining fluid balance, transmitting nerve signals, contracting muscles, and keeping organs in the heart and kidneys functioning properly. Even slight imbalances can cause significant clinical consequences. Understanding where electrolytes are located in the body and how water is compartmentalized is a key to interpreting lab values. For example, sodium is a major extracellular cation, while potassium is the primary intracellular cation. Disturbances in these distributions underlie many clinical conditions. Two-thirds of our total body water is inside of cells, and this is intracellular fluid. The remaining third is extracellular, divided into several smaller compartments, 25% interstitial fluid, 1 to 2% in transcellular spaces like cerebrospinal, intraocular, and serous fluids. And this distribution helps us understand shifts in fluid and electrolyte balance that occur in health and disease. Fluid balance is a dynamic process. The body continuously regulates input and output to maintain homeostasis. A neutral balance occurs when input equals output. If input exceeds output, we have a positive balance, leading to fluid accumulation. Conversely, when output exceeds intake, a negative balance occurs, which may result in dehydration. Both situations can be clinically significant and affect labs. Homeostasis of solutes and fluids requires these or relies on these balances. For example, in dehydration, we see an increase in sodium concentration because water loss exceeds sodium loss. In overhydration, sodium levels may be diluted. These shifts directly influence electrolyte levels and blood chemistry. So, interpreting labs requires us to always consider the patient's hydration status. Blood contains both electrolytes and non-electrolytes. Non-electrolytes are uncharged things, proteins, glucose, urea, etc., even gases. Versus electrolytes are charged particles that we can break into cations, which are positively charged, and anions, which are negatively charged. The balance of these solutes determines osmolality, electrical neutrality, and overall cellular function. Serum osmolarity or osmolality, on the other hand, if we remember all the way back to the beginning of physiology, measures the concentration of dissolved particles in blood or in the blood per unit volume. It helps us evaluate fluid and electrolyte imbalances and is particularly useful in conditions like seizures, ascites, dehydration, and acid-base disorders. It also helps us detect unusual solutes in the blood, things like organic acids, sugars, or even ethanol. And clinically, it's a quick way to assess hydration status and metabolic balance. The most important contributors to serum osmolality are sodium, glucose, and blood urea nitrogen, or BUN. The calculation is 2 times the serum sodium plus glucose divided by 18 plus BUN divided by 2.8. Normal serum osmolality ranges from 285 to 295 milliosmoles per kilogram. When levels go above 385, patients may experience stupor and seizures could occur. Around 400 to 420 and values above 420, these are often values that are fatal. This makes osmolality an important diagnostic and prognostic tool. And this is looking at our favorite picture in practice, showing our red blood cell when placed into different solutions. When osmolality is too high, we think of dehydration, hyperglycemia, or renal dysfunction. When too low, it suggests overhydration or SIADH. The key point is small changes in osmolality can have profound neurologic effects, since the brain is particularly sensitive to fluid shifts. This is also digging into kind of this process, looking at the role of solute balance in maintaining cell volume. Water moves across membranes to equalize concentrations, and if plasma osmolality rises, water leaves the cell, causing them to shrink. If plasma osmolality falls, water enters the cells, leading to swelling. Clinically, this is especially important in brain cells, where swelling or shrinking can produce neurologic symptoms such as seizures, confusion, or coma. Another component that we should consider is our aspect of fluid movement is its relationship to pressure gradients. Water and electrolytes move constantly between compartments in response to osmolality, hydrostatic pressure, and oncotic pressure. This dynamic exchange keeps tissues hydrated and nourished, and when disrupted, we see clinical problems like edema, dehydration, or volume overload. Four main mechanisms govern this fluid movement. Osmosis is water moving towards a higher solute concentration. Diffusion is solutes moving from high to low concentration. Filtration is driven by hydrostatic pressure, like blood pressure, moving water and solutes across a membrane. And active transport moves solutes against their concentration gradient and requires energy, such as with the sodium potassium pump. Together, these processes maintain homeostasis. It is important to remember that membranes that separates body fluid compartments are freely permeable to water, which means that while solutes may be unevenly distributed, water moves readily to balance the concentrations, which is why changes in these ions, like sodium, glucose, or proteins, can dramatically affect water distribution between intracellular and extracellular spaces. And this is just reinforcing the concept that electrolyte imbalances often present as fluid imbalances. For instance, hyponatremia, low sodium, is as much a water problem as it is a sodium issue. Understanding these underlying principles helps us to anticipate how a lab value will correlate with clinical findings, such as edema, dehydration, or neurologic changes. Speaking of edema, it results when too much fluid accumulates in the interstitial space. Four major mechanisms can lead to this: increased hydrostatic pressure, decreased plasma oncotic pressure, increased capillary permeability, or lymphatic obstruction. Each of these mechanisms can be linked to disease, things like heart failure, liver disease, sepsis, lymphatic blockages, and recognizing the mechanism will help us guide our treatment. Electrolytes then are minerals with electrical charges that are critical for organ function. They help to maintain fluid balance, nerve conduction, and muscle activity, including our heartbeat. Importantly, body fluids remain electrically neutral. The number of positive and negative charges must balance. Because if electrolytes become too high or too low, the consequences can be life-threatening: arrhythmias, seizures, even paralysis. And here are our key players that we measure: sodium, potassium, chloride, calcium, magnesium, and bicarbonate. It's important to know that magnesium is not on a comprehensive metabolic panel. It'd have to be added on as a separate order. Each of these electrolytes have their own special role, but together they maintain osmotic balance, acid-base balance, and neuromuscular function. And when we interpret these lab results, we rarely look at them in isolation. We're often comparing them together to understand the broader clinical picture. And this is showing kind of the relative concentrations of these ions. We'll stop here. When we come back, we will look at calcium.